Now if you break down the word carbohydrate, we have carb and hydrate. So carbo comes from carbon. Hydrate comes from water. So carbon. Whoops. No, I don't want you to move. I'll write over here. Okay. Some of my text boxes are too big, so they want to move when I touch them. Okay, I give up. Carbon and water. Water being made up of hydrogen and water. So this word comes from the fact that carbohydrates is made up of carbon, hydrogen, and oxygen. And carbohydrates exist as monosaccharides, disaccharides, and polysaccharides. So you need to look at the prefixes here. We have monosaccharide. Mono means one. Disaccharides. Di means two. And polysaccharides. Poly means many. So monosaccharides are made up of one molecule of a sugar. Disaccharides are made up of two of these monomers, two of these smaller molecules, and polysaccharides are made of many, many, many simple sugars. It could be in the range of thousands of simpler carbohydrates linked together into these huge complex polysaccharides. Some common examples of monosaccharides include glucose, fructose, which is fruit sugar. Some common examples of disaccharides include sucrose, and sucrose is just a glucose and a fructose put together, and this is your white table sugar. Lactose, that's milk sugar, and maltose is another one. And maltose is made up of a monosaccharide called galactose, and malt sugar is found in anything with malt in it, like molasses. The polysaccharides that we're going to be looking at include starch, cellulose, glycogen, and chitin, and I'll talk about each of those later. So this is the most common monosaccharide out there. This is glucose. And glucose has the chemical formula C6H12O6. You notice the ratio of carbon to hydrogen to oxygen is 1 to 2 to 1. And that's true of all carbohydrates. They always have that ratio. When you look at this structure, this is a ring structure, and what we do is we number the carbons off. So we start over here in the corner. This is carbon 1, then 2, 3, 4, 5, and 6. And we can see that there are a couple of different kinds of functional groups in the carbohydrate molecule. There are these hydroxyls. And up here on the 6th carbon, we have a carbonyl group. So we have hydroxyls, those alcohols, and we have carbonyls, or carbonyls. So these functional groups allow glucoses to have reactions with other carbohydrates and form these long chains or huge ring structures that we see in cells. Now the previous slide showed you glucose as a ring structure. In solution, glucose actually ionizes. So in our body, which our molecules are dissolved in solution, they're dissolved in water, they tend to ionize. So these ring structures will break up and form these linear chain molecules. So normally in our cells, glucose looks like this. Here's the 6 carbons again. You can see them in the chain with their hydroxyl groups, and here's the carbonyl group down here. If you look at fructose over here, count the carbons. 1, 2, 3, 4, 5, 6. 6 carbons. If you count the hydrogens, we've got 3 up here, 4, 5, 6, 7, 8, 9, 10, 11, 12. And count the oxygens. 1, 2, 3, 4, 5, 6. Remember, glucose was C6H12O6. So these two molecules have the exact same chemical formula. But if you look at their structures, it's a little bit different. It looks a little different up here. It's different here. It's the same on this carbon, same on this carbon, same on this carbon, and same on this carbon. But this end of the molecule looks different. So it has a different structural formula. Glucose and fructose are referred to as isomers. They're molecules with the same chemical formula, but they have a different structural formula. And we see this with the carbs quite a bit. Very similar compounds, but different structural formula. These two molecules have different physical and chemical properties as a result of these small structural changes. So this is really common in carbohydrates as well. So I've just shown you a couple of monosaccharides, glucose and fructose. I don't have a slide for the disaccharides. You would just put two of those monosaccharides together to make a disaccharide. And as I said earlier, sucrose is a common disaccharide. That's white table sugar. And that's the glucose and the fructose molecule put together. And you would put them together using a condensation reaction. So now we're going to take a look at the really interesting carbs, in my opinion. These are the polysaccharides. These molecules are made of hundreds or thousands of monosaccharides linked together. There are four main types of polysaccharides: starch, cellulose, glycogen, and chitin. The graphic below doesn't show you chitin, but it's quite similar to cellulose. So each of these little, well, hang on, I want to draw on you. It's not going to let me. Okay, I don't know why it does this sometimes, but anyways, each of these little hexagon-shaped molecule is a glucose. And you can see that starch has many of these glucoses linked together in a linear chain. Cellulose, also many of these little glucoses linked together in a chain. But what do you notice is different about cellulose's structure compared to starch? Hopefully you're seeing that every second glucose is flipped. It's upside down. And the reason is because of the bond between the glucoses. And I'm going to get to that in a future slide. But starch and cellulose are very, very different. Starch is the storage form of energy in plant cells. Cellulose is the structural component of cell walls in plants. So they're both found in plants, they're both polysaccharides, they're both made up of these long chains of glucoses, but this one little structural change between cellulose and starch gives that molecule a completely different purpose in a plant cell. Starch is for storage of energy, cellulose is for support around the plant cell in the cell wall. The third one listed here, glycogen, it's quite similar to starch, but you'll notice it has these branching glucoses coming off of it. Starch also branches, but it branches differently than glycogen. Glycogen is only found in animals. So this is how we store our energy. When we have excess energy from our food that we haven't burned off through our daily activities, we will store it in our muscles and in our liver as glycogen. And that's why I had a picture of Usain Bolt that was to give you the idea that we need to store energy in our muscles. This is the molecule that does that for us, glycogen. Chitin. Chitin is a polysaccharide found in the exoskeletons of insects and crustaceans, and that's why I had a picture of a crab, because it's a crustacean. That really hard, tough, rigid exoskeleton of those kinds of organisms is primarily made up of this polysaccharide called chitin. It's similar to cellulose, and it folds itself up and creates this sort of mesh-like structure that's very, very, very rigid. A crab would put minerals from the sea that it lives in into the chitin molecules and create this almost a bone-like structure. But the foundation or the skeleton of this exoskeleton is this polysaccharide called chitin. So what allows our monosaccharides, or monomers, to link together to make these huge polysaccharides is called glycosidic linkages. It's a type of bond, okay? It's a bond type. And we're going to be looking at about 4 different bond types throughout the study of the macromolecules. This is the first one. I would encourage all of you to, in your notebook, create a table of bond types in macromolecules. And begin with glycosidic linkages. So in your table, you'd want to have the name of the bond type, what it looks like, and what do you find it in. So glycosidic linkages are what link sugar molecules together or carbohydrates together to make larger carbohydrates. So this graphic is showing you a condensation reaction, and the condensation reaction is linking together two OHs on two glucoses. And we put two glucoses together, you make maltose. And the bond that's linking it together, we can see this bond right over here with the oxygen. That's called a 1,4-alpha, it's Greek letter alpha, glycosidic bond, or glycosidic linkage. And this is what accounts for a lot of our, the making of disaccharides and for a lot of our polysaccharides. And you see we have water over here, so there's also water formed in this reaction. So that's called a condensation reaction, and the bond that forms is a 1,4-alpha glycosidic bond. The 1,4 comes from the fact that, remember we numbered our carbons? So we have the number 1 carbon here, and on the second molecule we have a number 4 carbon. So that's where the 1,4 comes from. The alpha part of the name refers to the bond angle. So I don't know what this bond angle is with the oxygen here, but whatever it is, that's designated as alpha. In this graphic, again, we have that alpha 1,4 glycosidic linkage. It looks just like the previous slide. But if you look over here, our bond over here is between the 1 carbon of this sugar or monosaccharide and the number 6 carbon of the other monosaccharide. So this is referred to as an alpha 1,6 glycosidic linkage. Okay, so this is an alpha 1,4 and an alpha 1,6. In this graphic, I show you a completely different bond, similar but different. Again, here's our alpha 1,4 glycosidic bond, like we've seen already. But in this second graphic, to make lactose, it involves putting a galactose with a glucose. And the difference is right here in that glycosidic bond with oxygen, the bond angle is different. It's a little hard to represent this on the screen with a diagram because this is a three-dimensional molecule. If you could build this bond, you would see that the bond angle is different. So this is referred to as a beta. This is the Greek letter beta. Beta 1,4 glycosidic bond. Running out of room here. Glycosidic bond. Now, where is this relevant? Well, when we look at cellulose, cellulose has some beta 1,4 glycosidic bonds in it. And that is very relevant to us as people who need, we are animals who need to digest cellulose. We eat plant material. We need to be able to break down cellulose. The fact that cellulose has this beta 1,4 linkage is what prevents us from breaking down the cell walls of plant material. And you'll learn more about that as we go through the work in the textbook and a future lab that we'll be doing. So this brings us to the end of this video. You've seen an introduction to carbon and why it's important in biochemical molecules. You've learned what a functional group is, and we've examined some of the common functional groups. We've talked about the 4 main biochemical macromolecules that we'll be studying, and then we looked at carbs. So you learned about monosaccharides, disaccharides, and polysaccharides. You learned about how monomers can be linked together through condensation reactions to form larger molecules like disaccharides and polysaccharides. You learned about hydrolysis reactions, which will break down those larger carbohydrates into their simple monomers or their simple monosaccharides. And then you were introduced to some of the more common carbohydrates. So take some time to really try to make sense out of all this and read your textbook, watch videos, read your notes over, and we will continue to work with these biochemical molecules with molecular model kits and in a huge lab that you'll be doing soon. In the next video, we'll talk about the lipids.